Hello, everyone. I'd like to welcome you to the virtual Newhouse Center for the Humanities. I'm Eve Zimmerman, Director. Today, it's my distinct honor and pleasure to introduce Professor Natali Valdez to you. A member of the Department in Women and Gender Studies, Professor Valdez was a summer Newhouse Fellow in 2020, our COVID summer, and she has graciously offered to share her work with you today. Today is a special day, partly because I understand that we have some prospective Wellesley students in the audience, and I'd like to extend our warm welcome to you. Professor Valdez received her PhD from the University of California, Irvine in 2016. She was subsequently awarded a 2-year postdoctoral fellowship at Rice University, where she ran a faculty feminist reading group, furthered her research, and acted as a mentor to graduate students from groups not adequately represented in academia. Since coming to Wellesley, Professor Valdez has published work in top journals such as Medical Anthropology, co-editing and writing the introduction for a special issue on racism in reproduction. Professor Valdez asks a question of pressing importance to our ability to function as a society. Why do health disparities across race remain so glaring after 30 years of diversity and inclusion efforts in medicine? Her research interrogates the ways that pregnancy trials can adversely affect the very populations that they were designed to help. She poses an equally important related question as well. How, and I'm paraphrasing Natali, how do certain blind spots in science related to systemic injustice limit scientific creativity itself? Today, Professor Valdez will tell us about her cutting-edge research, which will soon appear in book form from the University of California Press. The title of the book is Weighing the Future: Race, Science, and Pregnancy Trials in the Post-Genomic Era. Keep an eye open. It is the first study of its kind. For prospective students in the audience, Professor Valdez teaches the Introduction to Women and Gender Studies. Yes, you can actually study with her when you arrive. She teaches a course on the body and a seminar on race, gender, and science, among others. She is a devoted teacher who takes her classes on enriching field trips, even via Zoom. Natali is also known as a tireless and compassionate community member who reaches out across all communities at the college. So a very, very warm welcome to prospective students and to Professor Valdez. Thank you.
Oh, thank you so much, Eve. That was such a wonderful introduction. So I will get started, and I have some slides to show, and then I hope to have plenty of time for some Q&A. So as Eve just described, my book is titled Weighing the Future, and I am excited that it will be hopefully coming out soon. So what I want to talk about today is I'll share a little bit about my book summary. I'll describe some of my methodologies. I'll define some key terms and concepts. I'll review the significance of this project and the key arguments, and highlight some key contributions, and then touch a little bit on some broader impacts. Okay, so what is this book about? Weighing the Future examines pregnancy trials and their socio-political implications. It is the first ethnography of ongoing pregnancy trials in the United States and United Kingdom. So the book is organized into 3 sets of couplets, and the first part really provides the historical and kind of definitions and origins. So what is epigenetics, what is postgenomics, and how have these fields integrated into child maternal health? And then chapter 2 kind of gives a little bit of a history of prenatal interventions that I study, which are lifestyle interventions of diet and exercise. And then part 2 starts to get into the ethnography, and chapter 3 explores recruitment practices and emphasizes themes of fatness, race, and risk. And chapter 4 really focuses on pregnant participants' experiences, and so I've called it pregnant narratives. And part 3, I explore environmental animation. So I try to address the question, what counts as the maternal environment? And in chapter 6, I review what data becomes valuable. So what kinds of pregnant biobits, which that's a term I use to describe the blood, the urine, the breast milk, sometimes feces. All of these pregnant biobits are collected during pregnancy trials, and they accumulate value. So in that chapter, I kind of look at why pregnancy trials are deemed valuable, even if they're not clinically proven to be to improve pregnancies. And I'll get into that a little bit further. Okay, so what are my methodologies? And here I describe my methodology as a feminist ethnography of science. And so what is that? Basically, I studied the scientists that design clinical trials. I observed and examined their methods and procedures, and I learned the languages of the field. And what this meant actually was to take 2 years of extra coursework in addition to my PhD to understand clinical trial design, child maternal health, and epigenetics. So my PhD is in anthropology, and I took these extra courses to understand how the scientists think, what kinds of terminologies they use, and how they design their work. And so what makes this project feminist? And primarily it's that I attend to politics of race, gender, and power. Okay, so what do pregnancy trials look like? Well, basically they apply the randomized control trial method, which has a control group, an experimental group, and gives an intervention to the experimental group and sees if their health outcomes improve as a result of that intervention. And so in this case, the interventions that are tested are lifestyle interventions of diet and exercise that are deemed safe for pregnant populations. So most pregnant people are not necessarily recruited for drug development, although that just changed. The NIH just changed their policy to include pregnant people into drug development trials. But for the sake of the trials that I'm talking about, no drugs are tested. It's mostly just behavioral interventions. So the target population here are overweight and ethnically diverse people. All of these trials are publicly funded. And just to give you a rough estimate, these trials can cost millions and millions of dollars. It's extremely expensive to run clinical trials. And most clinical trials are designed and funded in the United States. Actually, 75% of all clinical trials, not just interventions, behavioral interventions, but all, including drug trials, 75% of all clinical trials globally are funded and designed by the United States. So there is a huge uneven distribution in the production of clinical trials, and they're often framed as the gold standard of evidence-based medicine because they influence global health policy. So studying these clinical trials took 3 years to gain access, and I had to gain IRB approvals from 3 different institutions across 2 countries. Okay, so what does this look like on the ground? What does it look like to have a feminist ethnography of science on the ground? So I interviewed scientists. I also followed participants through their clinical trial journeys. And only in the United States did I have dual roles, which meant that while I was collecting my own ethnographic data, I also worked as a staff member on the Smart Start trial. And I was trained as an interventionist, and I worked closely with the intervention staff and principal investigator. At the US trial, the interventions are in charge of delivering the intervention to pregnant participants in the intervention or experimental group of the trial. So as an interventionist, I met with pregnant participants every 2 weeks for about 5 to 6 months of their pregnancy. We also had weekly meetings with the PI to troubleshoot any intervention issues. And these sessions gave me insight into the experiences and challenges of other interventionists and their assigned participants. So tracing the experiences of the women in the intervention during their pregnancy gave me an intimate view into their everyday routines, challenges, and social conditions. So why are pregnancy trials targeting ethnically diverse, overweight pregnant people? Well, one of the reasons has to do with the fact that new science or epigenetic research is suggesting that what pregnant people eat during pregnancy can impact the risk of diabetes and obesity in their children or potentially future children. Because in pregnancy, the pregnant body is framed as the first generation, the fetus is the second, and then the reproductive cells in the fetus are the third generation. So that's how it's imagined that pregnancy is a critical period for future development. And just a note on terms, I do use pregnant people and women interchangeably because not all pregnant people identify as cisgendered women. So what is epigenetics? In brief, epigenetics is the study of gene-environment interaction, regulation, and expression. It's part of the post-genomic paradigm or the post-genomic era in science, which is often dated to be around the early 2000s, turn of the century. And it is indexed by the end of the Human Genome Project, which found that genes alone do not explain the existence of human variation and development. So what does that mean? Basically, what it means is that our genes are not the blueprint of our lives or health and disease, and that something else accounts for phenotypic variability. So that means how disease manifests have a lot to do with environmental stimulus. And so that's why epigenetics is so important. It's because it has fundamentally changed our understanding of the environment. So through epigenetics, we can now understand all of these factors to be important in influencing our health. The environment in epigenetics includes everything from molecules around our genes to toxic chemical exposures, and it also includes social interactions, which becomes really important. So how does epigenetics matter in pregnancy? Epigenetic modification stimulated by environmental factors during pregnancy can impact maternal health, fetal, and infant health. And through epigenetics, the maternal environment becomes really important during pregnancy. And so the pregnancy trials that I study draw from the field of epigenetics to link pregnant people's behavioral choices like diet and exercise with future health risks. Contemporary science frames pregnancy as a critical period of development, like I said, because it encompasses multiple generations in one. So what counts as the maternal environment in pregnancy trials? Well, it turns out that in randomized clinical trials, mainly the only environmental factors that can be studied are individual pregnant bodies and behaviors. So here you'll see on the left side of your screen that this is this like maternal environment. And of course, this picture is supposed to denote the kind of idea that whatever the pregnant person eats, then that influences fetal development. So these are the ideas that are shaping the application of epigenetics in pregnancy trials. Okay, but you know, what happens to all of these other variables of the environment? Basically, they just start to disappear from the study. And really what becomes important, and I highlight social interactions here because social interactions include experiences of racism, which are rarely studied in clinical trials, let alone pregnancy trials. But what becomes the most important variable of the environment for pregnancy trials are diet and exercise. Okay, so part of the reason that only diet and exercise are part of the pregnancy trial intervention is that RCT method is not designed to examine multiple intersecting variables. And we know that these environmental factors are not isolated. All these variables intersect and they do not occur in a vacuum. But it is very difficult for the RCT method to capture how these variables intersect. So this is a big question. So why does this matter? Well, the environments, I'm trying to make the case in the book. Well, I do make the case in the book that the environments that shape genes, bodies, and future health and pregnancy go beyond individual bodies and behaviors. They include race, gender, and structures of inequity. For instance, pregnant participants in the clinical trials that I worked on are part of different social, economic, and cultural environments. Not all participants have the same choices or options to choose from. Many of the participants I worked with were unemployed, had unstable housing, and were financially struggling. For those participants, the challenge to buy fresh produce and take safe walks in their neighborhoods was much more, it was much more challenging for some compared to other participants. And in addition, different participants experience daily encounters of racism and anti-Blackness, and these experiences cause stress that can also impact fetal development during pregnancy. So individual lifestyle interventions are framed as if all bodies live in similar environments and have equal access to healthy opportunities, choices, and material conditions. And what I found is that this is not the case. So pregnant people classified as at high risk for diabetes and obesity are targeted for individual lifestyle interventions and are also embedded in racist and poor resourced environments that make it nearly impossible to comply with the intervention during the trial or sustain the intervention changes after the trial is completed. So importantly, focusing only on narrow definitions of the maternal environment does not promote effective maternal health policies because, like I mentioned earlier, most, if not all, of the pregnancy trials that test lifestyle interventions on individual pregnant participants have been inconclusive, which means that they haven't found significant proof that they improve pregnancies. And I have a couple explanations for why this might be, and it has a lot to do with how we think about our methods. So we know what the problems are. What are alternative applications of epigenetics in pregnancy studies? And in my book, I argue that a feminist and critical race framework can help us understand the maternal health problems and solutions differently. So what does this look like in practice? Okay, so traditional frameworks, many people will frame the problem as having to do with health disparities. Brown and Black people experience higher rates of obesity and diabetes because of their individual behaviors and lifestyles. So then if you frame the problem like this, the solution becomes change the individual behaviors of ethnically diverse populations. Okay, but that hasn't technically worked so far because of the inequalities in the material conditions that unequally shape people's opportunities and choices to make healthy decisions. So what can we do here? And what I'm proposing in the book is applying feminist frameworks to the problem. Okay, so let's reframe the problem. It's actually racist disparities. So it's not just health disparities across race, more explicitly, it's racist disparities. And there's a growing movement in public health that's trying to embrace this language because oftentimes if we use health disparities or inequalities in socioeconomic status, it obscures the fundamental power dynamics that are related to race and that shape many people's chances at employment, housing, education. And so racist disparities brings back the focus to these inequalities in a more direct kind of way. Okay, so in this framing, the problem then becomes Brown and Black people live in racist environments that make them sick. So then what can we imagine the solution to be? Intervene at structural levels to interrupt systemic and institutional racism. And I'm making the case that thinking about the problem, drawing from a feminist and critical race framework can help us not only change how we design clinical trials, but also their effectiveness and the ways in which we can develop health policy that efficiently addresses issues of equity and racist disparities. Okay, so what might be the broader impacts here? And one of the main questions that has come up in doing my research is, should RCTs be used to study environmental epigenetics? Because the environment is so complicated, it's so entangled. And if RCTs need individual discrete variables, and the environment can't really be studied as an individual variable, then potentially we need to rethink our methodologies. Okay, the other question is that, potentially we need to broaden the scope of the maternal environment, and doing so can promote different questions. So if we think about the maternal environment more than just individual pregnant bodies and behaviors, then we might think of how pregnant or parental leave policies shape reproductive health. And many feminist scholars of reproduction have brought so much attention to how fundamental labor policies are to reproductive health. Another question that emerges when we think more capaciously and less heteronormative about the maternal environment is we can start to think about how to be inclusive to transgender communities and child maternal health, which is a huge issue currently. So the other question that I want to touch on is how to more directly address issues of equity in clinical trial developments. And this is absolutely relevant to COVID-19. So in the last few minutes that I have, I'm just going to share with you some thoughts about this issue in relation to COVID-19. So while it is true that for the development of the COVID-19 vaccine, it was necessary to include diversity, what diversity actually means for pharmaceutical development relates to body size, ages, environmental exposure, and genetic variation. That may impact drug metabolism and response. Ethno-racial classifications of race as an index of diversity can be misleading. And scholars have explained, and this semester in my class, Race, Gender, Science, a seminar, which a lot of my students are here today, because it was required. But in that seminar, we had a guest lecture from a pharmacist, Dr. Alexa Volpe, who is assisting in the vaccine distribution in Texas. And she commented in the seminar that, quote, when researchers include race-related characteristics in their trial demographics, what they truly mean to say is that there is an inclusion of a diverse array of genetic variables that can influence drug metabolism and response. Yet, race is a poor proxy for genetic polymorphism. So, for example, many Latinx individuals can also use a classification as white for race. However, the Latinx population has one of the greatest amounts of genetic diversity. End quote. So Dr. Volpe is echoing well-established understanding that there's more variation within imagined race ethnicity group than between such groups. The important distinction to make is that Black and Brown people are needed in clinical trial development, not because they are Black or brown in skin pigmentation or an ethnic racial classification. Black and brown bodies do not metabolize drugs differently because they identify as a certain race. The narratives around diversity often tread a narrow line between, on the one hand, essentialism and deterministic biology, and on the other, liberal democratic notions of inclusion. All different bodies are needed in clinical trial research, and racial classification of difference is not biologically based, but rather socially, historically, and politically constructed. The point of highlighting this issue in the unfolding of COVID-19 is similar to the argument that I make in the book, which is that focusing too much on diversity and inclusion via proxies of race and ethnicity classifications often distract from effective or precise characterizations of diversity. So we need better understandings of diversity in science from, uh, on the one hand, that don't necessarily rely on unstable racial classifications. And the other reason why we need more rigorous understanding of this is that um, there are structures of racism that produce unequal health outcomes. And so this is the point of around equity. So on the latter point, the disparities of health across race that promote a more inclusive approach to evidence-based medicine in the late 90s. And what I'm talking about is that in 1993, the NIH made a mandate to include more women and racial minorities into clinical trials. This was their big effort to do inclusion, diversity and inclusion. So unfortunately, though, it hasn't actually unfolded in the ways that people have wanted it to. So that is, even if vaccine development included enough diversity, such approaches do not ensure that the vaccine is distributed or accessed in an equitable way. Based on data collected from the Census Bureau, the locations of healthcare facilities that can distribute vaccines are not easily accessible to poor, brown, and Black communities in the southern region of the United States. Medical infrastructures that exist in the US are predominantly in white affluent neighborhoods. Thus, through design and infrastructure, Black communities will and have had a hard time accessing vaccination. Besides the locations that are available for distribution, folks will need flexible employment to make appointments in different countries and to follow up in different counties and to follow up. Such requirements are challenging for people in precarious contract labor, seasonal farm workers, or in the service industry. Intervening on the unequal and racist conditions that shape people's lives and health require a distinct set of understandings of equity and diversity. And that is the case that I make in the book. And I will stop there and thank you so much for your attention.
Well, what do you say, Professor Valdez? Can I say how very grateful we are and how this is just opening a whole world and a bridge between the humanities center, what we do at the Humanities Center and what the more traditionally defined as social science departments do. And so I'm just really grateful because I see that as the future in a sense of both sides of the fence. And I think your book is going to be a major step forward in that. And, I have a lot of takeaways, maybe I'll put them in in an email to you. But thanks again. Thank you. Thank you.
Thank you.